Hello everyone. Today we are going to discuss some questions from the genetics and DNA sections. Okay, and I'm going to explain the concept also regarding that questions related to that questions. First question is nitrogenous base plus sugar is known as nucleosides, nucleotides, polypeptide, polysome, none. First, let me tell you what is nucleic acids. Nucleic acids are the polymer of nucleotides. And there are two types of nucleic acids. You must know. You all must be knowing regarding this. There are two types of nucleic acids. One is a DNA, and another is RNA. When you talk about the DNA, this is deoxyribonucleic acid, and this is ribonucleic acid. Okay. The basic difference between DNA and RNA is of the sugar. That is, in DNA, deoxyribose sugar is present. In this, ribose sugar is present. And out of 4 nitrogenous base, in presence of in case of DNA and RNA, in difference between DNA and RNA, there is a difference of nitrogenous base also that you will come to know. Now, if you talk about just I said you that this nitrogen, nucleic acid is a polymer of nucleotides. Okay. Now let's see. First, there are two types of sugar. You have known regarding the sugar. One is deoxyribose, and another is ribose. Then now you talk about the nitrogenous base. Then it is of two types. One is pyrimidine, nitrogenous base. In pyrimidine, you see it is there are 3 nitrogenous base: cytosine, uracil, okay, and thymine. But when you talk about the purine, it's adenine, and another is guanine. You must know that adenine forms hydrogen bond with the thymine, while guanine forms hydrogen bond with the cytosine. But it is in case of DNA. In case of RNA, adenine just forms bond with the uracil. This is the difference between DNA and RNA, not nitrogenous base. And another one is phosphate group. Then when sugar joins with the nitrogenous base, they form nucleosides. When phosphate group comes and gets attached to the nucleosides, it forms nucleotides. But our question is restricted to nitrogenous base plus sugar. Then answer will be nucleosides. Which of the following is wrong about genetic code? Okay, genetic code, there are codon, anticodon in that. Which code for the specific amino acids, and in turn amino acid is responsible for the formation of protein. The total 20 amino acids are necessary for the this formation of protein. Okay, then the question is asked about the genetic code, that which of the following is wrong about the genetic code. First is triplet in nature. Yes, of course, genetic code, yeah, codon, that is AUG, or any codon that is in triplet in nature. It consists of 3 nitrogenous base. It is universal in nature. It is applicable for the all types of the organism, whether it be unicellular or multicellular, or virus, protozoa, anything. Genetic code is same for all the organism. And it's just overlapping. Means suppose in one codon it is this guanine is present, but this means a guanine will be the part of both the codon AUG as well as GUC. Okay, according to this overlapping nature. No, but it's wrong, because if AUG, this guanine is the part of this codon, it cannot be part of another codon. Okay, means genetic code is of non-overlapping nature. Okay, this is wrong about genetic code. 2n-1 is which type of aneuoploidy? Okay, monosomy, double monosomy, nullisomy, trisomy, tetrasomy. See, and if you talk about the mutation, then there is a chromosomal mutation as well as gene mutation. In chromosomal mutation, either there is a change in the chromosomal number, or either there is a change in the chromosomal structure. When you talk about the chromosomal change in the chromosomal number, then either there is a change in the number of chromosome in a set, or there is a complete change of chromosome set. Okay, when there is a change in the number of chromosome set, that is euploidy, when there is a change in the number of chromosome in a set, that is aneuploidy. Suppose when you talk about the euploidy, then in euploidy you will see it's a monoploidy, then it's X, diploidy, then 2X, triploidy, then 3X. But when you talk about the aneuploidy, and in aneuploidy, suppose there is a loss of chromosome in a set, 2n is the set minus 1, means there is a loss of one chromosome in one set, then this will be monosomy. There is a loss of two chromosome, one from each set, minus 1 each set, then it will be double monosomy. Okay. Bar I character in drosophila is observed due to the which chromosomal abbreviation. Okay. Now I told you that chromosomal mutation can be of two types. One mutation is of change in the structure also. See, suppose this is the chromosome. Now see, this is the chromosome 1, 2, 3, 4. Now this is chromosome 3, 4. Means this segment of chromosome has been broken, and now this chromosome form, means this part has been removed. Now so here only 3, 4 part is left, means the terminal deletion of chromosome. Okay, this is terminal deletion. The question is bar I character in drosophila is observed due to the which chromosome abbreviation. Next is inversion. In inversion means there is a breaking of the chromosome segment, but the reunion in the same chromosome in reverse order. Okay, in reverse order means suppose this is a chromosome. I'm talking about this segment, the 5, 6, 7, 8. Okay, they break the chromosome, but now they becomes 5, 7, 6, 8. Okay, the reunion. Okay, and this affects the crossing over. In case of duplication, what happens when you talk about the duplication? See, suppose in case of duplication, A, B, C, D. Now what happens? It will become A, B. Now same chromosome A, B, C, D. Okay, then A, B, C, D, and this broken part will join here. This is duplication. Okay, and the bar I character in drosophila is due to this thing only. Okay, duplication. Helix length of DNA in angstrom is, you must know that suppose if you talk about the DNA structure, I hope you all know the structure of DNA. Double helical structure is DNA. Okay, then this is one turn. In one turn, this nitrogenous adenine, thymine, guanine, cytosine, this is combined. But in one turn, there are distance between two steps is 3.4 angstrom. Distance between two turn is 3.4 angstrom, and in one turn there are total 10 steps are there. Okay, 10 steps. Then what will be total length? 34 angstrom, and the length of complete one turn is known as the helix length. Then answer will be 34 angstrom. In one turn there are 10 steps. Synthesis of protein over RNA is known as replication, transcription, translation, mutation, and this you all must be knowing. See, suppose we talk about the DNA, RNA, and protein. Then if DNA is formed from the DNA, then it is a replication. When RNA is formed from DNA, it is transcription. When protein is formed over RNA, then that is translation. Okay, then the question was asked about translation. Okay, see, I have not gone deep into these questions. Otherwise, if I go deep into the expression, it will take a lot of times. But anyway, if you have any doubt, you can ask us through the message or comment in the box what you want. We'll make next video regarding that. I have already told about our various courses. If you all are preparing for the NABARD, you can take these courses, and this will cover all the agriculture parts that is covered in the syllabus of NABARD. And this course is of regarding AFO. There are 4 different types of courses in the AFO. Then you can visit the website and you can take according to your needs. Thank you. See you in next lecture.